All right, guys, to do this protein synthesis race, you're going to click Start a New Game and take a look at the description of transcription. Transcription is the copying of a gene, like a segment of our DNA, in order to make mRNA. It is the first process, the first step that we have to do in order to make a protein. So we have to do transcription by first unzipping our DNA molecule so that we can take a look at our individual strands. So we unzipped our DNA and now we have two separate strands. It says, notice that the DNA molecule has unzipped. Only one strand will be used for transcription. You're going to use the top strand, go ahead and click on it, and now look at our enzyme. An enzyme called RNA polymerase is going to be used to make the mRNA, specifically RNA nucleotides, that we need to build this molecule. So we're going to click on RNA polymerase, and we're going to use this enzyme to build me my RNA strand. So it says RNA polymerase is touching the first nucleotide of the DNA sequence. Find the RNA nucleotide that's complementary to that first DNA nucleotide. If there's a T in DNA, then RNA polymerase is going to bring an A. This is called transcription. So we're going to continue to add the nucleotides with the following complementary base pair rules. Now remember, if there's an A in DNA, there is no T in RNA to complement that. So instead, we're going to use U, uracil. But C still goes with G. Miss Culp is a cool girl. But A goes with U. We said always upbeat. C with G. T still goes with A. Right? A goes with U, etc. So we're going to follow complementary base pair rules and just continue down this strand. Okay, so now that I have finished moving down the DNA strand, it says transcription is done. You have made mRNA. mRNA stands for messenger RNA, and that is going to carry the message, aka the information that I needed to make my protein. So now that I'm done, DNA can zip back up to form that double helix, and the mRNA is going to leave the nucleus and go to the cytoplasm to find a ribosome, which is where we're going to actually make our protein. So we're going to click on the mRNA, and we're going to send it out toward the ribosome, which is out in the cytoplasm. Okay, so now the mRNA leaves the nucleus, which DNA can never do. It's going to head out toward the ribosome to help us make proteins. So that was transcription. We're going to put our DNA back together. It's going to wind back up to that double helix. We don't need any more. It can be transcribed again later if necessary. So, first question says, DNA and RNA are both made of subunits or building blocks or monomers called what? Well, DNA and RNA are nucleic acids, so they're both made of nucleotides. What is the name of the enzyme that adds new nucleotides, aka the building blocks of DNA and RNA, to build RNA? Well, RNA is made by RNA polymerase. DNA polymerase makes DNA during replication. RNA polymerase makes RNA during transcription. How many strands does DNA have? So think about what we started with. We had this double helix that we unzipped with two strands, but I only use one of them to make a one-stranded RNA molecule. Which mRNA nucleotide is complementary to guanine? Remember, G and C always go together. Miss Culp is a cool girl. Which RNA nucleotide is complementary to A, adenine? A still goes with, wait a minute, is there T in RNA? Don't think so. Instead, we have to use U, uracil. So Miss Culp is always upbeat. Which RNA nucleotide is complementary to T? T still goes with A. And what is the function of the RNA that we made? So mRNA is going to basically take this code, this information that's in our DNA, and we're going to transmit that code in order to make proteins out to the ribosome, which is the site of protein synthesis. We don't use mRNA to make tRNA. We make tRNA in the same process called transcription. And we are going to use mRNA to carry all that information that we needed out of the nucleus to the ribosome. Oops, sorry. So we're going to click both of those. Is correct, A and C. What happens to DNA once transcription is done? So now that I'm done making the mRNA, we are going to zip it back up until we are going to need to transcribe that gene again. All right, so now that we did transcription, we're done, we did it. We're going to move on to the last step of protein synthesis, which is called translation. So now it asks us to move the mRNA to where it needs to go. So if you guys look at these little holes, they're called pores in our nuclear envelope. And we can have the mRNA leave through those little holes, those pores, and go to the ribosome. The ribosome is where translation will occur because that's where proteins are made. Ribosomes make proteins. So now in order to understand translation, we need to know that to build a protein, we're basically just linking together or bonding together amino acids. The order of the amino acids in our chain and the length of the chain is determined by the mRNA, and it will determine what kind of protein we're making. So the mRNA is going to carry these instructions for us on how to make the protein, using codons. A codon is a triplet, meaning 3 nitrogen bases, 3 of those letters. And each codon codes for one specific amino acid. For example, the codon AUG codes for this amino acid called methionine, shortened as MET, and you guys can see that in this codon chart. So let's look at the mRNA. The first codon is located in the P site, aka right here on the ribosome. What is that first codon? AUG, we're going to click on that because remember, a codon is 3 letters. So we're going to find the tRNA with the complementary anticodon. Anticodon is the complementary basis, 3 letters, to the codon in mRNA. So if AUG is our first codon, then UAC is our anticodon. So now I can move that tRNA with my arrows, and we're going to pick up a specific amino acid that he's going to bring in order to build this protein. So we can use this little codon chart over here to figure out what one he's going to bring. And if you look up AUG as our codon, he's going to bring MET, the blue guy. Okay, so we're going to move him around. Oh, look, he already picked it up for me. I was right there. And we're going to bring it to match the codon in the mRNA. So the tRNA is going to bring the amino acid. It's carrying the amino acid. It's transferring it, which is what the T stands for. We're going to repeat this process with the rest of these codons until I build this protein, aka a polypeptide. So again, let's look at our next codon. UCA is the codon, the 3 letters in mRNA. UCA codes for serine, the little red amino acid. Now we need to find the anticodon to get the right tRNA molecule. So if UCA is the codon, then AGU is my anticodon. And we said that he's going to pick up a red amino acid. So look, as you can see, we just bonded those 2 amino acids together. It's called a peptide bond, and that's the start of building my polypeptide. Let's do one more together, and then I'll skip through it. AUG, again, I'm going to need UAC as my anticodon because it's just the complementary 3 bases. I need to pick up a blue amino acid. You can see that in the codon chart. AUG codes for MET. We're going to bring that here. And again, I'm just building this chain of amino acids. So UCA is my next one. AGU, same guy, is going to be my next amino acid in this polypeptide. All right, so I continued to move down the mRNA, looking at each codon, matched it with the appropriate anticodon in the tRNA, picked up the appropriate amino acid based on my codon chart, and I built this polypeptide. So I just finished translation. Now the 2-part ribosome separates, and we basically release what we've made. So notice that I have this chain of amino acids. Each amino acid is connected to the next amino acid next to it with a peptide bond. So we call this chain of amino acids a polypeptide, since poly means many and peptide is the type of bond that we've used. This is kind of like a basic protein. Now we want to fold it and process it and make it into the correct shape, because the shape of a protein is what gives it its function, right? Shape equals function. So it says identify the part of the arrow or bracket that we're pointing to by clicking on the correct word. All right, so for example, I'm pointing to this strand right here. This thing that I used with all the codons on it is called mRNA. 3 letters, 3 bases on mRNA is called a codon. That's what we're going to use to determine what amino acids will be brought to the ribosome. This little molecule with the weird swirly thing at the top and an anticodon on it is called tRNA, or transfer RNA. 3 bases or 3 letters on tRNA is called an anticodon. And the tRNA is picking up these little circles in our animation, which are just amino acids, the building blocks of proteins. Where do we do this? Where do we do translation? I do it at this site right here, which is a ribosome. and that is the location of this process. So, what is translation? The making of a protein using the codons and mRNA as instructions, making mRNA using DNA, making tRNA and mRNA and ribosomes, or producing RNA polymerase. Translation, which is what we just did, is making proteins using mRNA codons, aka the instructions from mRNA. What does the mRNA do? It is the instruction, it's our template that we're using to know what amino acid should be next in our sequence. tRNA specifically is bringing amino acids. He was the guy who was carrying those little circles to the ribosome in the correct order. The codons are on mRNA. mRNA has codons, anticodons. I think the T in anti helps me to remember tRNA anti. So tRNA has anticodons, which are just the complementary bases to the codons in mRNA. What happens at the ribosome? I make proteins. It's called protein synthesis. mRNA codons and their complementary anticodons and tRNA match up. Amino acids are joined together through peptide bonds to make a polypeptide, aka like a protein. So all of these things are true. Amino acids are the monomers of proteins, like the building blocks. They are not the monomers of anything else. They just make up proteins. A polypeptide, remember poly means many? A polypeptide is basically like a protein, okay? Essentially like a baby protein is what I call it, because it could be like processed and put in the right shape first. Essentially, we just peptide bond a bunch of amino acids together, and it's what we're making in translation. So all of these are true. And that is translation and transcription. So protein synthesis involves two steps: transcription, which is using DNA to make mRNA, and then translation, which is using the mRNA to figure out the amino acid sequence and bonding those amino acids together to make a polypeptide, aka a